In the mid-20th century, the discovery of DNA's structure established the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. It established a rigid, one-way street of biological information. Treating DNA as a passive, read-only template leaves a massive gap in our understanding. Every cell in the human body holds the exact same genetic code. A static blueprint cannot explain how a single sequence produces vastly different tissues, or how a body dynamically adapts to stress in real time. To understand how this works, we have to look at the physical reality inside the nucleus. DNA is not free-floating. To fit 2 meters of genetic material into a microscopic space, the double helix is tightly spooled around dense protein clusters, compacting into a solid, impenetrable structure called chromatin. The static sequence of bases is insufficient on its own. There must be an active, higher-order physical mechanism dictating which parts of the code are actually accessible at any given moment. This mechanism is the epigenome, a regulatory layer of chemical modifications sitting on the DNA sequence. Studying epigenetics forces us to stop thinking about genetics as abstract information and start looking at the gritty, physical chemistry of the nucleus. The genome is an interactive structural machine. By physically dictating genome accessibility, the epigenome acts as the precise mechanical interface between external environmental inputs and our genetic output. The foundational unit of this physical packaging is the nucleosome. Each nucleosome consists of a core made up of 8 histone proteins, with the DNA strand wrapped tightly around it. The biophysics here rely on charge. The DNA phosphate backbone carries a negative charge, which binds tightly to the positively charged lysine tails protruding from the histone proteins. To express a gene, the cell adds an acetyl group to a lysine tail, neutralizing its positive charge. The negative DNA strand is repelled, drifting from the core to create euchromatin. This gap allows bulky chromatin remodeling complexes like SWNF to wedge in. Only after this clearance is achieved can RNA polymerase bind to the strand and initiate transcription. Gene activation relies entirely on the mechanical shifting of molecular obstacles to expose the raw genetic code. The cell also deploys mechanisms that act directly on the DNA strand itself to shut genes down. The primary targets for this repression are CpG sites, regions with high concentrations of cytosine and guanine base pairs, typically located at the promoter regions of a gene. DNMT1 patrols promoters, attaching a bulky methyl group to bare cytosines. This creates steric hindrance, physically barricading transcription factors from docking. The methyl tag recruits MBD proteins and deacetylases, stripping open marks to pack the area into dense, impenetrable heterochromatin. A separate protein, UHRF1, recognizes these specific methylation marks and ensures they are meticulously copied across every cell division. While acetylation temporarily unpacks the genome for active use, methylation locks it shut, establishing highly stable, long-term gene silencing. We can observe these microscopic mechanics scaling up by looking at how skeletal muscle responds to acute physical exercise. The mechanical stress of a heavy workout triggers a massive release of intracellular signaling kinases, which flood through the cell and breach the nuclear membrane. Once inside the nucleus, these kinases act as catalysts, activating local histone acetyltransferases. These enzymes execute a highly targeted strike, chemically opening the chromatin specifically at the SLC2A4 gene locus. With the physical barrier removed, RNA polymerase docks at the locus and begins transcribing the exposed gene into messenger RNA. That mRNA travels out of the nucleus and is translated into GLUT4 transport proteins, significantly increasing the muscle fiber's capacity to absorb glucose. An external environmental stressor directly rewires the cell's metabolic phenotype by mechanically prying open a specific genetic locus. Zooming out to the lifespan of a human body, our cells constantly alter these chemical marks in response to decades of nutrition, toxic exposure, and psychological stress. Researchers discovered these specific DNA methylation patterns accumulate and decay at quantifiable rates over a lifetime. This animated scatterplot demonstrates an epigenetic clock. It tracks DNA methylation changes, comparing the accumulation of marks against strict chronological age. But the trendline doesn't capture everyone. Lifestyle factors cause biological age to drastically diverge from the calendar. Because the epigenome records the cumulative physical toll of an organism's environment, it serves as the most accurate biomarker for true biological age. For over a century, classical inheritance models operated on a strict assumption: acquired environmental traits die with the organism. The mammalian body actively enforces this rule. During reproduction, germline reprogramming aggressively erases the vast majority of epigenetic marks, providing the offspring with a blank genetic slate. Yet, specific anomalies exist. Through a process called transgenerational epigenetic inheritance, specific environmental adaptations can successfully bypass this erasure and pass intact through the germline. We see this in historical human cohorts. Following the severe starvation stress of the Dutch famine, descendants of survivors exhibited permanently altered DNA methylation marks multiple generations later. This systemic, inherited memory sharply contradicts the mid-century model of DNA as an unyielding, static blueprint. If the genome serves as the organism's hardware, the epigenome acts as its real-time operating system, compiling external environmental data into a physical, biological reality that echoes across generations.